Have you ever really thought about how flowers, being stuck in one spot and all, manage to have a secret sex life? It sounds a little wild, doesn't it? But honestly, it's one of the most incredible stories in all of biology. Today, we're going to pull back the curtain on exactly how flowering plants reproduce. And I promise you, by the end of this, you will never look at a simple flower the same way again.
I mean, seriously, think about it for a second. Plants can't move. They can't walk around. They can't go on dates. They can't just swipe right to find a meat. They are literally rooted to the spot. So how on earth do they solve this massive, massive problem? Well, the answer is as beautiful as it is brilliant: the flower. So let's dig in and see how these incredible, intricate structures make it all happen. So here's our game plan for today. We'll start by figuring out what a flower is actually for. Then we'll dive deep into the nitty-gritty of how they make the male and female gametes. After that, it's all about matchmaking, how they get those gametes together through pollination and fertilization, and what happens next. We'll even look at some of the weird and wonderful ways certain plants break the rules. Sound good? Let's get into it. All right, first up, why do flowers even exist? Sure, they're pretty, they smell nice, and we love them for that, but that's not their main job. From the plant's point of view, a flower is a high-tech reproductive machine, an incredible piece of engineering built for one thing and one thing only: making more plants. And get this, the whole process starts way before we ever see a single pretty petal. Deep inside the plant, it has to decide, okay, it's time to flower, time to reproduce. A whole cascade of hormones kicks in, triggering structural changes, and this tiny little bud called a floral primordium starts to grow. And that right there is the beginning of the flower, the stage where all the magic is about to happen. So imagine we're building a flower from the outside in. First, you've got the calyx, which is made of sepals. These are those little green leaf-like parts that protect the bud before it blooms. Next, you add the corolla, made of petals. This is the show-off part, all bright and colorful, designed to basically shout, "Hey pollinators, over here." But those are just the accessories. The real action, the essential reproductive work, happens in the middle. You've got the androecium, which is the male part, and right in the very center, the gynoecium, the female part. That's where the real drama unfolds. Okay, so let's zoom in on that male part, the androecium, and specifically the stamen, to see how it makes the male gametes that are going to get this whole reproduction party started. Think of a stamen like a tiny matchstick. The long, slender part is the filament, and the head of the match is the anther. And that anther? That's the pollen factory. If you look closely, you'll see it has two lobes. That's what bilobed means. And each of those lobes has two chambers, or theca, making it dithecous. So 2 lobes, 4 chambers in total, all just cranking out pollen. Now, if we were to slice open one of those little pollen factories, a microsporangium, we'd see it's protected by 4 distinct wall layers. The outside layer is the epidermis, kind of like its skin. Just inside that is the endothecium, which has a super important job. It helps the anther burst open to release all that pollen. Next are the middle layers, which are sort of temporary support staff. And finally, the innermost and arguably most important layer, the tapetum. This is like the kitchen and the nursery rolled into one, providing all the food and nutrients for the developing pollen grains. It's so dedicated that its cells are packed with dense cytoplasm and often have more than one nucleus just to keep up. So, what's going on inside this protected chamber? Well, right in the center, we have a group of cells called the sporogenous tissue. As the anther grows, these cells gear up for a special type of cell division called meiosis. Each one of these cells is a potential pollen mother cell, or PMC. And this whole process, making tiny microspores from a pollen mother cell, is called Microsporogenesis. I know, it's a bit of a mouthful, but the crucial thing to remember is that because it's meiosis, each diploid mother cell splits into 4 tiny haploid cells which stick together for a while in a little group called a microspore tetrad. All right, pop quiz time. This is a classic question, so let's see if you've been following along. Can you put these 5 terms in the correct order of development? Take a second to think about it. Got it? Okay, the correct sequence starts with the sporogenous tissue, which is full of pollen mother cells. Those mother cells undergo meiosis to form the microspore tetrad. The microspores then mature and separate to become individual pollen grains. And finally, inside those pollen grains, the male gametes are formed. How'd you do? If you didn't get it, no worries at all. Just rewind a bit. That's the logical flow. As the anther dries out, those little microspore tetrads break apart, and each one develops into a pollen grain. And these things are built like tiny tanks. A pollen grain has a 2-layered wall. The outer wall, the exine, is made of this stuff called sporopollenin, which is basically indestructible. Seriously, it can survive ridiculously high temperatures, strong acids, you name it. We haven't even found an enzyme that can break it down yet. It's so tough, it's the reason we can find pollen in fossils that are millions of years old. The inner wall, the intine, is a bit more delicate. It's a thin layer made of cellulose and pectin. So how long does this super tough pollen actually last? Well, it really depends. For some plants, like rice and wheat, the pollen is only good for about 30 minutes after it's released. But for others, it can last for months. And here's where it gets really cool. We can actually freeze pollen in liquid nitrogen at a bone-chilling minus 196 degrees Celsius, and it will stay viable for years. We have these pollen banks all over the world, which are just like seed banks. It's an amazing tool to preserve genetic diversity and help us breed better crops. All right, we've seen how the male gametes are made, packaged, and preserved. Now it's time to check in on the female side of things. Let's head back to the center of the flower, to the gynoecium, and see what's happening there.
So the gynoecium is the female part of the flower. Sometimes it's made of just a single unit, a pistil, which we call monocarpillary. But other times there can be a whole bunch of them, making it multicarpillary. And when there's more than one, they can either be all separate and independent, which is apocarpus, or they can all be fused together into one solid structure, which is syncarpus. It's kind of like the difference between a bunch of individual grapes versus a single solid pineapple.
Each individual pistil has three main parts. At the very top, you have the stigma, which is like a sticky landing pad designed to trap any incoming pollen. Then there's the style, which is the long tube connecting the stigma down to the swollen base, the ovary. And inside the ovary, you'll find the ovules, which, if all goes well, are the future seeds.
So let's take a closer look at one of those ovules. This diagram is a must-know for exams, so pay attention. The ovule is attached to the ovary wall by a little stalk called the funicle. The main body of the ovule is wrapped in a couple of protective layers called integuments. They cover the whole thing except for a tiny little opening at the top called the micropyle. Think of it as a little doorway. Inside all of that, we have a mass of cells called the nucellus, which is like the pantry, packed full of food. And right in the middle of it all is the main event, the embryo sac. That's where we'll find the female gamete, the egg cell.
So how do we get from that simple ovule to a fully formed embryo sac? It all starts with a process called Megasporogenesis. One of the cells in the nucellus, usually near that micropyle doorway, gets chosen for a special job. It grows large and becomes the megaspore mother cell, or MMC. This is a big, important cell, packed with dense cytoplasm, ready to do some heavy lifting. And just like the pollen mother cell, it's going to go through meiosis. Now, that megaspore mother cell splits via meiosis into 4 haploid megaspores. But here's where things get really interesting and, frankly, a little brutal. In most flowering plants, only one of those 4 megaspores actually survives. The other 3 just wither away and degenerate. The plant puts all of its resources into nurturing that one chosen megaspore. And because the entire embryo sac develops from just that single surviving megaspore, we call this monosporic development.
So let's put Microsporogenesis and Megasporogenesis side by side to really nail down the differences. The location is different. Anther for micro, ovule for mega. They both use meiosis, but the big, crucial difference is the outcome. In microsporogenesis, all 4 microspores are functional and become pollen grains. But in megasporogenesis, it's like a botanical Hunger Games. 3 degenerate and only 1 functional megaspore survives. It's a key distinction that comes up a lot.
Now, let's follow that one surviving megaspore on its journey. The nucleus of the megaspore divides by mitosis 3 times in a row. So we go from 1 nucleus to 2, then to 4, and finally to 8 nuclei. But here's the weird part. These divisions are free nuclear, meaning the cell itself doesn't divide, just the nuclei inside. So you have one big cell with 8 nuclei just floating around. It's only after that 3rd division that cell walls finally form. The result? A very specific structure with 7 cells, but 8 nuclei. Don't worry about the math, just remember it. It's a classic exam question. Okay, so our male gametes are packaged and ready to go inside the pollen grain, and our female gamete is waiting inside the embryo sac. But there's one huge problem. They're in completely different places, and neither of them can move. So how do they get together? That's where pollination and fertilization come in. It's time for the grand union. Pollination is basically the plant kingdom's long-distance matchmaking service. It's just the transfer of pollen from the anther, where it's made, to the stigma, the receptive landing pad. This single act is the critical first step in bridging the physical gap between the male and female parts. Without it, fertilization is just not going to happen. So how does the pollen actually travel? Well, plants have basically outsourced this job. Some use things like the wind. This is called anemophily. They make tons of light, non-sticky pollen and just hope the wind carries some of it to the right place. Others, like aquatic plants, use water. That's hydrophily. But most flowering plants are much smarter about it. They use animals, which is called zoophily. They make themselves all pretty and smelly and offer up nectar to attract bees, butterflies, birds, even bats, to act as their personal pollen couriers.
Now, just because a pollen grain lands on a stigma doesn't mean it's game over. The pistil is very, very picky. It has to recognize if the pollen is from the right species. This recognition is a complex chemical dialogue between the pollen and the pistil, called pollen-pistil interaction. It's like a secret handshake. If the pollen is the right type, the pistil accepts it and lets it grow. But if it's the wrong kind, it gets rejected. It's an amazing little quality control system.
So, once a compatible pollen grain gets the green light, it germinates. It grows a tiny pollen tube down through the style all the way to the ovule. This tube carries 2 male gametes. It's like a tiny Trojan horse, delivering its precious cargo right to the heart of the female territory. And when it gets there, it releases those 2 male gametes into the embryo sac. This sets the stage for the climax of the entire story, an event unique to flowering plants. Double fertilization. You're going to want to pay very close attention here. This is one of the most important topics for your exams. So what exactly is double fertilization? It's 2 fertilizations happening at almost the same time. The first one, called syngamy, is when one of the male gametes fuses with the egg cell. This creates the diploid zygote, which is the baby plant embryo. The second event is called triple fusion. This is when the other male gamete fuses with the 2 polar nuclei in the large central cell. This creates something called the primary endosperm nucleus, or PEN, which is triploid, 3N. So you have one fusion to make the baby and another fusion to make the baby's lunch. It's a brilliantly efficient system.
So, the deed is done. Double fertilization is complete. But what happens now? This is where things really start to transform. We're now entering the post-fertilization stage, where we'll see how that zygote grows into an embryo, how the PEN turns into a packed lunch, and how the entire ovule and ovary transform into a seed and a fruit. Right after that double fertilization, 2 major things start to happen. The zygote develops into the embryo, and the PEN develops into the endosperm. But here's a crucial rule of plant biology. The endosperm always develops first. And why? Because the endosperm's only job is to be the food source for the growing embryo. It's like the plant is making absolutely sure it has a nutritious lunch packed and ready to go before the baby embryo even starts to develop. Pretty smart, right? So let's just quickly recap the incredible transformation that happens after fertilization. The zygote becomes the embryo, the primary endosperm nucleus becomes the endosperm, the integuments, those protective outer layers, harden into the seed coat, the entire ovule matures into the seed, and at the same time, the ovary ripens and becomes the fruit, with the ovary wall becoming the fruit wall, or pericarp. All the other parts of the flower, the pretty petals, the sepals, the stamens, their job is done and they just wither and fall away.
So that's the standard playbook for most flowering plants. But of course, nature loves to experiment and always has a few tricks up its sleeve. Let's take a look at some of the interesting ways that plants sometimes break the rules. First up, we have something called Apomixis. It sounds complicated, but it's basically asexual reproduction that pretends to be sexual reproduction. These plants make seeds, but they do it without any fertilization happening. It's a way for the plant to make little clones of itself, all packaged up as seeds. And this is a really big deal for agriculture. If we could figure out how to make our hybrid crops do this, farmers could save and replant their own seeds year after year. It could be a total game changer for food security. Next up, we have Polyembryony. And just like it sounds, it means many embryos. This is when you get more than one embryo developing inside a single seed. You might have actually seen this if you've ever planted a lemon or an orange seed and 2 or 3 little seedlings popped up from the very same seed. It's basically like having twins or even triplets, but for plants. All right, we have been through a lot today, the entire journey from flower to fruit. So before we finish up, let's do a quick rapid-fire recap of the most important things you absolutely need to know for your exams. Okay, these are the non-negotiables. You need to be able to draw and label that anatotropous ovule in your sleep. You need to understand how that weird 7-celled, 8-nucleate embryo sac is made. You absolutely have to master double fertilization, and critically remember the ploidy levels. The zygote is diploid, 2N, and the PEN is triploid, 3N. Know the different agents of pollination and their examples. And finally, be able to define Apomixis and Polyembryony. If you know all of that, you are in fantastic shape.
So the next time you see a flower or you pick up a piece of fruit to eat, I want you to just pause for a second. Think about everything that had to happen to make that possible. The journey of that tiny, indestructible pollen grain, the incredible efficiency of double fertilization, the complete transformation from a flower into a fruit. It's a pretty amazing story, isn't it? Does it make you appreciate the natural world just a little bit more?
And that is a wrap on the secret sex life of flowers. I really hope this deep dive helped make this complex and super important chapter a little bit easier to understand and remember. Thanks for joining me. Keep up the fantastic work with your studies, and good luck. You've got this.